Hello, today we're going to be talking about Chapter 13, which is the regulation of gene expression. And we're going to be talking about that in prokaryotic cells and eukaryotic cells, because there is a little bit of a difference. So first, let's talk about the prokaryotic regulation. So remember, prokaryotic cells are bacteria. They have no nucleus. They're simple life forms. Bacteria do not require the same enzymes all of the time. Their environment is always changing, so their enzymes are produced only as needed. So we're going to be focusing in on the operon model. In particular, we will be talking about the lac operon. This experiment, that Jacob and Monod did, showed E. coli is capable of regulating the expression of genes. Okay, so an operon is a group of structural and regulatory genes that function as a single unit. And structural genes are genes for a metabolic pathway. They're grouped together on a chromosome and they're transcribed at the same time. Okay, so to go along with the operon, it consists of three different things. Okay, the promoter, operator, and then the genes. Okay, the promoter is the DNA sequence where RNA polymerase first attaches, and it is a short segment of DNA. It signals the start of the operon. It begins transcription of those group genes. The operator is the DNA sequence where active repressor binds, and it also is a short segment of DNA. When the active repressor binds to the operator RNA, polymerase cannot attach and transcription cannot occur. Okay, and again, the structural genes are the ones that code for a metabolic pathway, and it's a little bit longer segment of DNA. The regulatory gene is a gene that codes for the repressor protein. It's normally located outside the operon, and it controls whether the operon is active or not. So it's kind of like a switch. So first, let's talk about the trip operon. Okay, the regulator codes for a repressor. All right, and then there's two things. There's a thing if tryptophan is present or if it's absent. So this trip operon is a pathway for the synthesis of the amino acid tryptophan. Okay, so if tryptophan is absent, the repressor is unable to attach to the operator, and the expression is normally on. The RNase polymerase will bind to the promoter, and enzymes for the synthesis of tryptophan are produced. Okay, if it's present, it will combine with the repressor protein as a co-repressor. So tryptophan is kind of like a co-repressor. The repressor becomes functional when bound to tryptophan. Repressor blocks the synthesis of enzymes in the pathway for tryptophan synthesis. Okay, so this is a repressible operon. This is operons involved in the anabolic pathways that make things. And here are some animations that will show you all this. So this is the trip operon. You can see where the operator is, the promoter, the regulatory gene, the structural gene is. So when the repressor binds to the operator, transcription is prevented. Okay, and the top one is when tryptophan is absent. So the enzymes needed to synthesize tryptophan are produced. And that's when the repressor is unable to attach to the operator, so RNA polymerase will bind to the promoter, and then it will make the tryptophan. The bottom picture is when tryptophan is present. The presence of tryptophan prevents production of enzymes used to synthesize tryptophan. So it combines with the repressor protein as its co-repressor. The repressor becomes functional when bound to tryptophan, and the repressor blocks the synthesis of enzymes in the pathway for tryptophan synthesis. So now we're going to talk about the lac operon. Enzymes that break down lactose are encoded by three different genes. There's a gene that codes for B-galactosidase, which was going to break down lactose into glucose and galactose. There's another gene that codes for permase, which facilitates the entry of glucose into the cell. And then there's a third gene, transaxlase, which is an accessory function in lactose metabolism. All right, so the lac operator is a regulator that codes for the repressor. And again, there's two options, if lactose is absent or if lactose is present. So if lactose is absent, okay, again, the milk sugar, the repressor attaches to the operator and the expression is off. If it's present, it combines with the repressor and renders it unable to bind to the operator. RNA polymerase will bind to the promoter, and then those 3 enzymes that I just talked about are necessary for lactose catabolism are produced. All right? So lactose is an inducer of the lac operon. So this is called an inducible operon, which is the lac operon. And this usually is found in catabolic pathways that break down a nutrient. So again, we have 2 different options. We have A and B. So again, when you look at this, you have your operator, your promoter, your genes, and you have your RNA polymerase and your repressor. So the regulator gene codes for the lac operon repressor that ordinarily binds the operator and prevents the transcription of the 3 genes. All right, well, there's further control that goes along with this lac operator. E. coli preferentially breaks down the glucose, and lac operon is maximally activated only in the absence of glucose. So when glucose is absent, CAMP, or cyclic AMP, accumulates, and that's going to bind to the catabolite activator protein, which is CAP. Then, bound to CAMP, it will bind to a site near the lac promoter. When CAP is bound, or the catabolic activator protein is bound, RNA polymerase binds better to the promoter. Those structural genes of the lac operon are expressed more efficiently. All right, so CAP is the catabolic activator protein. Okay, and when it bonds to the DNA, it exposes that promoter to the RNA polymerase. Okay, now if glucose is present, there's little CAMP, cyclic AMP, in the cell. This relationship exists because the transport of glucose into the cell inhibits the enzyme adenyl cyclase, which produces CAMP. So that makes CAP inactive, and the lac operon is not expressed maximally. So we just have some more pictures of what's happening. This is when lactose is present and glucose is absent, and there's a lot of CAMP. All right, again, you can see what's happening here. And that's when the RNase polymerase binds. Okay, so the top picture is when lactose is present and glucose is absent, and you have a lot of CAMP, cyclic AMP, present. And the bottom picture is lactose present and the glucose is present, so then the CAMP level is low. All right, so now we're going to talk about the eukaryotic cells. Eukaryotic cells are a little bit more complex, so there's a variety of mechanisms, okay, that's going to regulate gene expression. And we do have 5 primary levels of controls. We have nuclear levels, cytoplasmic levels, and each of those will get broken down further. So the nuclear levels is chromatin, transcriptional, post-transcriptional, and then the cytoplasmic is translation and post-translational. So nuclear levels pertain to the nucleus, okay? Chromatin structure deals with the chromosome packing, and that is all dealing with genes being turned on, all right? Transcriptional control is going to be helping make the mRNA. Post-transcriptional control is when we process or shorten the mRNA. Now, cytoplasmic levels are pertaining to the cytoplasm. So, translational controls when translation occurs. And then post-translational control occurs after protein synthesis. So, again, this is just showing you both of that happening in the cytoplasm with the DNA to chromatin, then to the making the mRNA, and then out into the cytoplasm for protein synthesis. All right, so chromatin structure. This is pretty much chromatin packaging. And this is going to regulate the accessibility to DNA. All right, so we know that DNA is associated with histone proteins. It's bound tightly together to make up chromatin. And we've already talked about nucleosomes. This is when DNA is wound around a group of 8 molecules of histone, and it kind of looks like beads or pearls on a string. We call each of the pearls or beads a nucleosome. The levels of chromatin packing are determined by the degree of the coiling within the nucleosome. And that's just showing you how that occurs, how that will regulate gene expression. The nucleosomes present access to the DNA, so transcription cannot take place. If the histones are acetylated, then the access is achieved. If the tails are methylated, then the access is more difficult. All right, euchromatin is the loosely coiled DNA. That's the active DNA. That's what transcription is going to be occurring on. And then the heterochromatin is the tightly packed DNA, which typically is called inactive because transcription does not occur. Okay, and that is inaccessible to RNA polymerase. We also have bar bodies. Females do have two chromosomes, two X chromosomes, okay, where males only have one. One of them is typically inactive. So one chromosome is tightly packed along the entire length and is inactive, does not produce any gene products, and that's going to be called the bar body. So we have some examples for this. All right, this is in female cats. So the allele for black coat is on the X chromosome, and the allele for an orange coat is on the other. So patches of black and orange in coat can be related to which X chromosome is in the bar bodies of the cells found in the patches. Okay, so now we're going to go to the transcriptional control. This is very critical. This involves the participation of transcription factors, activators, and repressors. Okay, so transcription factors are proteins that help regulate transcription by assisting the binding of RNA polymerase to the promoter. All right, a transcription activator binds to the region of DNA called enhancers, so it's called the DNA binding protein. All right, so when it binds the enhancer DNA, that's one region of the DNA where factors that regulate transcription can also be found. All right, this activation occurs when they are phosphorylated by kinase. And this is just showing you a little bit. Okay, transcription requires the transcription factors binding to the promoter, transcription activators binding to the enhancer, okay, and then the mediator protein and DNA loop act as a bridge to join the activators to the factors. Next, we have post-transcriptional control. Okay, this includes alternate mRNA splicing and controlling speed at which the mRNA leaves the nucleus. So this is dealing with the mRNA. Okay, so once you have your mRNA, you're going to remove the introns and splice back the exons. Remember, you're putting on the methylguanine cap, poly A tail, and so forth. Okay, when introns are removed from the pre-mRNA, differential splicing of exons can occur, which affects gene expression. An exon normally included in the mRNA can be skipped and removed instead. This is just showing you a little picture of that. Okay, translational control, determines the degree to which mRNA is translated into a protein product. Okay, and this is going to kind of affect whether translation occurs and how long the mRNA remains active. Now, microRNAs regulate translation by causing the destruction of mRNAs before they can be translated. So these are small RNAs that can bind to and damage the translation of mRNA in the cytoplasm. Our last one is post-translational control, and this is going to affect the activity of the protein product. So proteases are enzymes that break down proteins. They're regulated on how long a protein remains active and are confined to lysosomes or proteasomes. And it is accomplished by regulating the activation and degradation rate. If you have any questions on this, please let me know. Thanks.